Okay, so our next speaker is going to be Blake Sweeney, who is surprisingly also for Amber, and he'll be talking about RNAcentral. And you want to say it yourself?
Hello, everyone. I'd like to thank the organizers for giving me the opportunity to speak here. My name is Blake Sweeney. I'm a bioinformatician at RNAcentral, and I'll be describing our services. I'll just be describing the services we provide. I should apologize. The links didn't copy over in the right color, so they're all probably impossible for you to see. Sorry about that. But our website's rnacentral.org. If you put in RNAcentral in Google, you'll find it. So what is RNAcentral? We are a database that collects all non-coding RNA sequences of all biotypes from all organisms, provides a single integrated view of them. So currently we have over 30 different databases. They span all sorts of things like genomic resources, things you may have heard of like Ensemble and HGNC, other model organism-specific databases like Flybase and Wormbase, linkRNA databases. Most recently, we've added LinkBook. This figure is slightly out of date. We have databases which are specific to various RNA types, Silva for rRNA, MERBase for microRNA, and so forth. And then we include archival sites like ENA and RFEM, which is a very general database. So with all of that, we provide this nice website here. We have three main features that we talk about: our text search, our sequence search, and our Genome Browser. But before I get into any of those in detail, I'll just tell you, you can batch download all our data. So we have an FTP site which allows download like you would expect. We have a REST API which powers our website. And we also now provide clones of our production database. So everything that I'm showing you, all the data, all the features, you can get yourself by querying our database. If you have any problems with that, please just ask me and we'll help you out. In theory, you should be able to run your own RNAcentral locally using the production clone, though we haven't tested it yet. So I'll start with the text search, which is built off of the great work that EBI does, that you just heard. So here it is. It looks pretty much exactly like what you just heard, but with our theming. It shouldn't surprise you, so it works the same way. Let's just look at a particular example. So I've looked at human telomerase RNA component, TERC. And this is a very zoomed out picture. I'd be impressed if any of you could read it. So I'm just going to walk through kind of the major sections and show you our features. So at the top, we provide an identifier. This is a unique stable ID for each individual sequence. So every sequence has its own identifier. This is very useful if you're creating annotations and such, like Go terms. We also tell you where the sequence has been observed, so if it's been observed across multiple databases, I'll let you know that. The other often underappreciated but feature I'm very proud of is we provide a human readable name. Often these names need to be computed from the given metadata that we have because people disagree about what something should be called, or they come up with nonsensical names, and we fix that in an automated way. We also provide interaction data. So tarbase and linkbase give you microRNA protein and microRNA long non-coding RNA target information. And we display that. In this case, this is a long non-coding RNA. And so we show you what microRNA is targeted. If were we looking at a microRNA, we would show you what proteins and long non-coding RNAs it targets. The next thing is a genome browser. So we align all sequences from an organism to their genome. So in this case, the top section is from Ensemble. So there are 2 Ensemble transcripts at the top, but then if you look in the boxed region, there are 8 RNAcentral transcripts. These are sequences which were not found in Ensemble. Likely they're found in ENA or somewhere else, which we have aligned into the genome and shown where they belong. This is something that kind of confuses people at RNAcentral occasionally, because we are a database of RNA sequences, not genes, so effectively individual transcripts. So sometimes people will ask for, what's the sequence of, say, TERC? And we can point them at, well, there are these 8 possible ones in this region. HGNC being a human annotated thing, you should likely use that. But there's apparently observed variation over what TERC is. We don't make a claim as to which one is correct at the moment. That is not something that we do. We are working to move to a gene-centric organization, so all of these entries would be collapsed into one thing, which is Turk, and then we would select one or a few as sort of the ones you should consider. But this is something to be aware of when looking at sequences in RNAcentral. We use ensemble compare to show related sequences if present. This example doesn't have them because they don't compute it for long non-coding RNAs, because no one really knows how to do that. We are considering ways of extending this related sequences thing, but it's into the future. We also show gene ontology annotations. So Go terms, if you're annotating a non-coding RNA, you're actually supposed to use RNAcentral identifiers because they're stable and unique to each sequence, so you can be very precise as to what's being annotated. So it's very easy for us to display these annotations. This one happens to have several of them. We generally only show manual annotations, and so some more high-quality things, which is very useful if you're looking for function. Finally, we do analysis of all of our sequences using RFAM, which is a database of non-coding RNA families, so not just individual sequences, but a whole family of related ones. If you're interested in RFAM, I recommend that you go to poster T58 tomorrow, I believe it is, and talk to Joanna, my colleague who works there. Also, while I have everyone's attention, RFAM is hiring a curator. So if you or someone you know would like a curator position, please get in contact. We are hiring. The EBI is a great place to work. I'm happy to talk about it later as well. Anyway, once we analyze sequences with RFAM, we can display, what family the sequence matches, if any. There is a Turk family, and pleasingly, this sequence matches it, so it gives you some confidence that this sequence is believable. We also use these RFAM annotations as a QA step, which I will show you later. The final thing we do is we show publications. Currently, these are only the ones we're given, so we aren't very extensive with our publications. This example is kind of intentionally bad. I could have found one which is great if I really wanted to, but I chose this because it shows that we only know what we're given at the moment. We are working towards doing a better job working with groups who do text mining as well as possibly doing our own text mining approach. So this is back to the sequence search. So I kind of glossed over it because you just saw Youngmi's nice presentation. But I do want to point out some of the neat features that we have at RNAcentral that other services don't provide. So this QA thing I mentioned, so you can go into the QA facet, which is collapsed, and go and look for something. And I chose a kind of a nice, simple example to show you guys. So this is a Rose non-coding RNA, supposedly. If you go into ENA and you pull down all the Rose non-coding RNAs, you would get this. However, we've analyzed it with RFAM and found that it actually matches a bacterial ribosome, at least partly. So it's not likely to be a Rose non-coding RNA. It's probably contamination in the sequencing experiment. In practice, you can find this in every database in every organism. I've seen, well-regarded databases have long non-coding RNAs that are actually fragments of bacterial ribosomes. I've seen well-studied model organisms that have riboswitches found only in bacteria, but somehow it's in the genome of this organism, and in no other eukaryotic organisms. So it's very useful to have because there are mistakes just inevitably, and this helps pick them up. QA is one of the things that we're planning to extend in the future. For example, look for protein coding domains and so forth in sequences. Now, I want to point out what's probably one of the more exciting features we just added to RNAcentral. So if you look at the text search facet, and you see this little section here called secondary structures. So now we have secondary structures for sequences. In fact, we have over 3 million secondary structures. If you're familiar with RNA secondary structure, you would think, well, where did you get 3 million of them? That doesn't exist. They're very painstakingly found experimentally. But what we do is we compute them, actually, and we display them in this layout. So this is a human small subunit drawn in the standard format for human small subunits. If you are also an aficionado of RNA secondary structure, which I kind of am, you will recognize this as the canonical way of drawing small subunits. So what we do is we go to the Gutel Lab, the comparative RNA website, where they have hundreds, over 800 diagrams of secondary structures. We build models for all of those, and then we use all those models to analyze every single rRNA, and then use that to determine both its base pairing information, so what's often called secondary structure, and then draw it in this nice way using a program called Traveler, which can use a template and draw a secondary structure in it. So the benefit to this is that if you are familiar with rRNAs, you can understand this. I can, in fact, show you a nice example from this one. So if you see those green and blue letters up at the top that are falling in this kind of circular loopy region, well, if you have studied rRNAs, you would know that that is a kink turn. That is a loop, that is a motif commonly found in RNAs, which induces a bend into a helix, because a kink and a turn, thus its name. It can tolerate a fair amount of sequence variability to form a kink turn. And it's really nice in that, this sequence, which had some variant from some other thing and you don't know what it means or what it does, when it's drawn in this way, you can zoom in and you can see, oh, that's a variant in a kink turn, that's sitting in this motif, I know these things about this motif. It's very useful. It's something we're very excited about. Currently, we only do it with rRNAs. However, I have just finished prototyping doing it with all RFEM models. And so about 80% of RNA sequences in our RNAcentral match an RFEM model. And so that means about 80% of these sequences will get one of these secondary structures drawn in the standard layout, which people are used to interpreting and understanding. We're very happy about that. Okay, so now I'll show you the sequence search, which Youngmi already mentioned. So, like, we have a sequence search. Every database has a sequence search, right? You put in your sequence, you get results. And they look like this. I've collapsed the alignments for easier readability. Then you'll notice on the left all these facets. These facets are wonderful. Not only do they allow you to drill down, like Youngmi pointed out, they're actually a visualization of your results, right? This is a summary table. So you know the biotype distribution of your sequences just looking at this, because it's the RNA type in the upper left. You have a good idea about the phylogenetic diversity because you see the organisms. Now, it's not a perfect display, and we're working on fixing that, improving that, and making it better. But it's a very simple thing, and it works pretty well to start with. So we're going to improve it as time goes on and use better methods of displaying things. And you can also use it to drill down, right? Sequences don't just fall from the air. You probably sequenced something. So in this case, if you know it was a human sequence, you can click human, and it's quite reasonable to say, well, I don't care about things that don't occur in the human genome. That's likely some sort of mistake or some issue. And so once you select, human found in the genome, then you see, oh, okay, this is a Turk. I happen to use a fragment of the original example I showed as the sequence search. Oh, wow, I'm way ahead of time. And so with that, I would like to stop and thank everyone involved. So this is a picture of the consortium that powers RNAcentral. We have our next consortium meeting in November, I believe, and so we're looking forward to that. If you're a database that works at RNAcentral and you haven't signed up to come, please get in contact with me. We can still get you in. I'd like to thank BBSRC for funding, EBI for giving us a home, and all of the RNA services team as well. Thank you.
Thank you very much for your work. And I have a first question is about if you search for microRNA targeting information, and I would like to know how did you get this targeting information? Is experimentally verified or just get it from prediction tools?
So link-based and tar-based are experimentally verified target information. We should be displaying the. Oh, we don't display it on this. If you go the other direction, so microRNA and what does it target, we actually show you the method that was used. So it might be, say, a luciferase assay, and we would say luciferase assay. If it's some more high throughput method, we would show you the name of it that was used.
And is it possible to add some filter, like if I just want the targeting for human beings,
Yeah, so sort of you can do a search that says all human microRNAs which are targeting this sequence or whatever, or all human microRNAs which have targeting information, and you could get that. Technically, there are some limitations on how much of that we can redistribute, because link-based and tar-based have a license that basically says you're not allowed to redistribute our data without our permission. So I have to be a little coy as to what you can and can't do with it.
Okay, like this data probably is based in like mere tar-based?
Yes. No, it's, I don't think it's the same as in mere tar-based. Yeah.
How did you gather this information?
Oh, so the two databases, link-based and tar-based, provide it.
Okay.
Yeah.
Okay. Thank you.
Thank you. I'm not an RNA person, but I'm certainly intrigued by your comments about the quality assurance problem, the contamination. I'm just wondering what the strategy is to clean things up. Off the top of my head, could you run background tasks that would take snippets of bacterial DNA and see if they could find matches in the wrong species and then try you to get rid of them?
Yeah, that's an interesting thing. So we only align sequences to the right genome. So it says it's human, we align to human. But we could try aligning human to bacteria, and then if it matches, say, that's a bad thing. We haven't quite thought about that. We're planning to do all against all searches. So if a human, take all human long encoding RNAs and run them against everything else. So if they started matching bacterial ones too well, that would actually be a good quality assurance step. We hadn't thought of doing that. That's a good idea.
Way around, if the bacteria is the common contaminant, you start with the bacteria and search, and then if it turns up lots of non-bacterial, you think there's something wrong.
Yeah, that's a good idea. We hadn't thought of using it that way, but that's good.
Okay, so I think our questions synchronized. We'll wait until, let's say, 11.